So Julieta, you have 15 minutes. Good luck with your presentation.
Thank you, Tim. Good morning, everyone. Today I'm going to talk about marine benthic invertebrates and ocean acidification, insights from a natural carbon dioxide vent in Galapagos using a fatty acid biomarker approach. This study is in the context of a collaboration between Ghent University and ESPOL, an Ecuadorian university. So ocean acidification is a decrease in the ocean pH due to increased atmospheric carbon dioxide. And as you can see by this graph, the increase in carbon dioxide in the atmosphere is also followed by an increase in the concentration of carbon dioxide in the water, which leads to a decrease of the pH. The pH has dropped 0.1 units since the Industrial Revolution and is estimated to further decrease between 0.3 and 0.4 units by the end of the century. Ocean acidification has many effects on organisms, including their physiology. And this is where fatty acids come in. Fatty acids are very important molecules. They are the main components of the cellular membranes. They're also the main building blocks of storage lipids. And they also have other roles such as molecular signal, pigments, and vitamins. In addition, they can be classified according to the degree of unsaturation or the number of double bonds. Here we have saturated fatty acids with no double bonds, monounsaturated with one double bond, and polyunsaturated with two or more. This last category, PUFAs, are particularly important because some of them can almost exclusively be synthesized by primary producers, which means that the consumers need to acquire them through their diet to meet their physiological optimal needs. And these are called essential fatty acids. And it's been seen that a lack of these essential fatty acids leads to negative effects in survival, growth, and reproduction. So fatty acids are important determinants of food quality, in particularly some parameters such as the ratio of PUFA to saturated fatty acids, and certain essential fatty acids such as EPA and ARA. These are have important roles in the immune system in organisms, and DHA that has an important role in regulating the membrane fluidity. But how does this relate to ocean acidification? Well, some studies show that high levels of partial pressure of carbon dioxide alter the fatty acid composition of primary producers, therefore altering the fatty acid composition of the consumer with negative effects for performance and fitness, and that these changes at the base of the food web can further be upscale the trophic web through consumption with potential changes for community structure and ecosystem functioning. However, most of these studies have been carried in laboratory conditions, and this imposes limitations on the time span of experiments. And natural carbon dioxide vents provide a natural setting where to test the long-term effects of low pH in organisms. This is a picture of the carbon dioxide vent in Galapagos, and these bubbles coming up are bubbles of carbon dioxide. And long-term exposure allows also to test for acclimation and adaptation of the response to the stressor. And also very important is that it takes into account species interactions and species-environment interaction that are also very important in mediating responses to stressors. Also, in other vents in the world, other organisms have been found living there. And fatty acids can tell us at what cost these organisms are living there. So the first objective of this study is to compare the fatty acid composition and total content of the sea urchin Euscidaris galapagensis between a carbon dioxide vent and control sites in the Galapagos Islands. And the second objective is to compare the fatty acid composition of the macroalgae with that of the sea urchin. And the associated hypotheses are that the fatty acid composition and total content in the sea urchin will differ among sites, and that the fatty acid composition of the sea urchin will mirror that of their food source. So a survey was conducted in the Galapagos Islands. These are the sampling points. Up here, Roca is the vent site where the vent site is located, and this is a volcanic islet, and down here is the carbon dioxide vent. And the other two sites, Albermale and Floreana, are the control sites. As you can see, the pH at the control sites is that expected for current atmospheric levels of carbon dioxide, while that at the vent is markedly lower if we take into account that the pH is a logarithmic scale. This is the pencil sea urchin, Euscidaris galapagensis. It's the most abundant sea urchin in the island. It's very important in structuring benthic communities because it feeds on algae, corals, and barnacles. 3 individuals were taken in each side of approximately the same size. Gonads were extracted, since gonads are where the lipids are stored and therefore fatty acids. And also gonads have shown to reflect the fatty acid composition of the diet. Additionally, 5 sea urchins were included from the Bent in a previous campaign in the same season in 2018. While algae were sampled opportunistically, here on the bottom you can see the algae communities were mainly encrusting coralline red algae, and in the bottom in the right you can see the grazing marks of the sea urchin on the algae. Additionally, samples of other important organisms of benthic communities were taken. We can see some gorgonids, black corals, and hermatypic reef-building corals. However, for these organisms, a fatty acid profile description was made, and this is included in thesis, but in this presentation I will focus on the sea urchin results. So for fatty acid extraction and analysis, gas chromatography was used, and a specialized software was used to identify and quantify the fatty acids. Regarding the data analysis, relative abundance concentration of fatty acids were used, and multivariate analysis were used to test the side effect on the overall fatty acid composition, while univariate tests were used to test the side effect on essential fatty acid, total fatty acid, and fatty acid classes. So these are the results regarding the first objective. This is a non-multidimensional scaling plot, and points that are closer together have a more similar fatty acid composition than points that are further apart. And here the R value goes from 0 to 1, and it tells you how similar or dissimilar are the sites. And 0.35 is rather low, so we can say there's not a strong site effect. However, in the plot, we can see that the control sites are more clustered together compared to the vent site that has higher variation. Regarding the total fatty acid content, there's no significant side effect, even though the controls have slightly higher levels, it's no significant. And regarding fatty acid classes, here what we can see is that shared letters, like here, represent no significant differences, while different letters represent significant differences. So for PUFAs, polyunsaturated fatty acids, the control number 1, albermale, was different from all the rest. And saturated fatty acids were higher in the vent compared to control 1. And the PUFA to saturated fatty acid ratio was higher again in the control 1, albermale, compared to all the other sites. Regarding essential fatty acid classes, again, we see that control number 1, albermale, has higher levels of arachidonic acid here on the right, and EPA compared to the vent, but in different years. So in arachidonic acid, control 1 is different from the vent in 2018, and in EPA is different from the vent in 2019. While for DHA, what we observe is the contrary. The vent in 2018 has higher levels of DHA compared to the control 1. And regarding the objective number 2, what we can see here is the fatty acid profiles for algae. The first 3 columns are different types of algae, and the last column is the sea urchin. And the y-axis represents the relative contribution of each fatty acid to the total. And what is interesting is that the fatty acids that contributed the most in algae were also contributing the most in the sea urchin. So the 14-0 and 16-0. EPA, the essential fatty acid mentioned before, and arachidonic acid. So there's a resemblance between the profiles of algae and sea urchins. So there's different mechanisms by which pH can affect fatty acid composition. As I mentioned before, it can be via diet. So it's been observed before that low pH changes the fatty acid composition in algae through different mechanisms of saturation and desaturation pathways. But it can also alter the fatty acid community and therefore changing the fatty acid composition as well. It's been also shown that sea urchins can cope with low pH, but this produces a change in energy allocation because there's a more energy demand for acid-base equilibrium at the expenses of energy storage. Yes. Also, pH can alter the lipid biosynthesis by altering the metabolism and gene expression. This has been observed in larvae of sea urchin and also in other organisms. And finally, gonads are reproductive organs, and as such, there can be differences between males and females, and this has been observed prior to spawning in other species of sea urchin. However, we do not know the reproductive cycle of this species of sea urchin, and we didn't assess sex, so we do not know this, but this could explain some of the variation. However, it is hard to establish the causes of the differences found, since there can be other factors also that have an effect, such as nutrients, chlorophyll, A concentration, and temperature. And this can also perhaps explain why the control 2 here in the south didn't show any differences with the bent, as control 1 did show. So one of the possible reasons behind this is that Galapagos is located in a system that has very dynamic system of currents. So here we have the Cromwell current and brings cold, nutrient-rich water. We have the Panama current bringing warm water from the north, and we have the Peru current bringing cold water from the coast of South America. And this creates very different oceanographic features in the islands, with gradients in temperature and nutrients from northwest to southeast. And also, at the beginning, the objective of this study was to also test the side effect on other important organisms for this community, such as gastropods and cnidarians. But this was not possible because the samples were very heterogeneous among sites, and fatty acid profiles are very species specific. So for these groups, instead, a fatty acid profile was reported, and this was included in the thesis. So in conclusion, the question of the effect of natural ocean acidification on the fatty acid composition of the sea urchin, Eucyaris galapagensis, macroalgae, and other macroventous invertebrates still remains. And to address this question, future research needs more replicates, also a more thorough understanding of the environmental parameters in the sites and of the local carbon chemistry. And also, it would be very interesting to link the fatty acid composition to other parameters such as growth, reproduction, and other metabolic parameters, and also to measure the consequences for energy transfer in food webs. Thank you very much. I want to thank my supervisors, Marlene de Trog, Rafael Bermudez, and Carl van Collen, especially Marlene de Trog for the close following and supervision, but also for her following during the coronavirus lockdown. Also Bruno in the laboratory for all the patients and teaching me about fatty acid analysis. My family and friends at home and here for all the support and motivation. Erasmus Mundus for the financial support, without which I couldn't be here. Also IMBRC for this opportunity and all the help through these years. And the team back at Galapagos for all the good work and good vibe in the sampling campaign. Thank you very much.